Welcome to today's biology class. In today's lesson, we are going to discuss digestion in humans. Okay, so the first thing first, let us define what digestion is. So digestion is the breaking down of food. That is what we call digestion. It's just a breaking down of food. Now, food can be broken down in two ways, and therefore there are two types of digestion. Okay, so what are the types of digestion that we can look at? We have physical digestion and chemical digestion. Let us start with physical digestion and look at what physical digestion is. So physical digestion is a breaking down of large pieces of food into smaller ones. So for example, let's say you are eating cassava. So you bite or you ingest, then you start chewing. That is breaking down into smaller pieces. Okay? And this is carried out by the action of teeth that is in the mouth. Okay? So physical digestion, we cannot just talk of the one that occurs in the mouth, but also some other section of the digestive system. But what you should know is that the breaking down of large pieces of food into smaller ones, and it is carried out by the action of teeth in the mouth. Other sections, we are going to look at that when we get there. Okay, and the other thing that you should know about physical digestion is that it does not involve enzymes. So very important point. So in the case, you might find questions in multiple choice where you're asked to differentiate physical digestion and chemical digestion. One of the differences that physical digestion does not involve enzymes. Now, what is the importance of physical digestion? So the importance of physical digestion, one of the importance of physical digestion is that it increases the large surface area, or it increases the surface area of the food for more efficient enzyme activity. Okay? So that enzyme can easily act on the food and further digest it. Then it makes food easy to swallow. So one of the importance of physical digestion is that it makes food easy to swallow. I guess you've not missed anything here. Then let us look at chemical digestion. Since we said there are two types of digestion, that is physical digestion and chemical digestion. For chemical digestion is a breaking down of large molecules. Pay attention, molecules of food into smaller ones. So in the physical digestion, large pieces of food are broken down into molecules. And these molecules, at the size of molecules, they are large. They need to be broken down into smaller ones. So chemical digestion is the breaking down of large molecules of food into smaller ones by enzymes. So the difference, physical digestion does not involve enzymes. Chemical digestion involves enzyme. Physical digestion is a breaking down of large pieces of food into smaller ones. Chemical digestion is a breaking down of large molecules of food into smaller ones, and by the action of enzyme. I guess you get the differences that are there between the two. All right, since we've looked at the importance of physical digestion, it's very important also that we look at the importance of chemical digestion. Therefore, what is the importance of chemical digestion? So it makes food easy, or it makes the absorption of food more efficient. Chemical digestion results into the food nutrients being broken down into the end product that can easily be absorbed into the bloodstream. So it makes the absorption of food more efficient. So that is the importance of chemical digestion. All right, so having looked at this, let us look at the human digestive system. Since we are going to look at digestion in human, it is very important that we familiarize ourselves with the human digestive system. And this is very important where exams are concerned, because just from the start, you expect questions in an exam. So it's very important that you know the parts of the digestive system and you know the functions. So at the moment, we are just going to look at just to look at the diagrams where these parts are positioned. But with the functions, we are going to look at the functions as we get to discuss digestion of the food nutrients at different stages of the alimentary canal or the digestive system. So one of the part of the digestive system is the mouth. Then we have the esophagus, as you can see is right here. We have the liver, although this one is accessory organ, it just helps in the digestion. We have the gallbladder here. These parts you need to know them. You have the stomach. You have the stomach. You have the small intestines. You have the large intestines. These parts, the pancreas, the liver, the bladder, all these plays an important role in the digestion, and therefore you need to know them. I mean to know where they are found in terms of labeling. If you are given a diagram, are you able to identify these parts? That is what is important. Are you able to tell what their importance in when it comes to digestion of food? That is very important question. So those are the things that you look at. So these parts that I've circled, including these that I've ticked, you need to know them. So just knowing these, you can answer at least 2 or 3 questions in an exam, because some of the question that comes is just a matter of identifying the parts. Okay? All right. So let us look at now the digestion of the food nutrients. So the food nutrients, the macronutrients that we are going to deal with, we are going to look at carbohydrates. So these are the ones that we are going to trace now. Proteins, as well as lipids. So these are the ones that we are going to discuss in terms of digestion. Okay? Now, let us look at the digestion of carbohydrates, proteins, and lipids. How are these digested? So now, digestion occurs in the mouth, or digestion starts in the mouth. Then it goes to the stomach, duodenum, and jejunum. So these are the portions that we are going to focus on. Okay, let us start with the first part, that is digestion in the mouth. So after ingesting the food, digestion starts in the mouth. Okay, so after food has been ingested, the following occurs in the mouth. So when you put food in the mouth, what happens is that you start chewing. And that is what another word that you can use for chewing is mastication. Okay, so we've started with the digestion of food nutrients in the mouth. Okay, so when you ingest the food, you eat the food. What you're going to do first is you start chewing the food. Now, what is chewing? Chewing is just a breaking down of large pieces of food into smaller ones by the action of teeth. So you use teeth to chew. Okay. Then of course, what follows as you are chewing, the process of chewing, you stimulate, or it results into the secretion of saliva. So there's a secretion of saliva also in the mouth. And now this is where now you need to pay particular attention, because we are going to look at the component of saliva and look at the function of each component of saliva. So secretion of saliva is carried out by the salivary gland. We are not going to go into detail to look at the salivary gland that you find in the mouth, but instead this one we are going to leave it for another part. Okay? So what are the components of saliva? So in the saliva you find water, and now we are going to look at each of these. What is the importance of water in the saliva? Or what is the role of water in the saliva? So the function of water, that it helps in cooling food too hot and warming up food too cold, so that its temperature is favorable. Remember where we are coming from. We discussed, or we looked at enzymes. Enzymes work well at a certain temperature. So when you eat too hot food, it has to be the temperature has to be lowered to a temperature where the enzyme work well. Okay? Then when it is too cold, still the food has to be warmed. So that is the function of the water in the saliva. That is helps in cooling food too hot and warming up food too cold, so that its temperature is favorable for enzyme action. That is one of the functions. The other one is that it softens food for easy chewing. Just imagine you put a biscuit in your mouth. What happened? It melts, and that way it is easy for you to chew and also to swallow. So saliva softens the food for easy chewing. Then in the saliva you find mucus. Okay, mucus. What is the function of mucus? The function of mucus is just to lubricate the food for easy swallowing. So it makes the food slippery so that it can slide well on the esophagus. So it lubricates the food for easy swallowing. What about salivary amylase? So the other component of the saliva is what we call salivary amylase, which is an enzyme. Okay, salivary amylase, which is an enzyme. Now, what is the function of this enzyme? So it starts the digestion of starch or carbohydrates in the mouth. So it converts or it breaks down starch to maltose. Now, remember we are looking at 3 food nutrients. That is the carbohydrates or starch. Let me put starch, proteins, and lipids. So in the mouth, digestion of starch starts. Okay, but since the food does not stay in the mouth for a long time, only about a percentage of starch is digested. Let me say 10% of it is digested in the mouth, which means the other percent is carried to other portion of the alimentary canal or the digestive system. So please, the enzymes, that is salivary amylase, start the digestion of a starch to produce maltose. Okay, and please, something that you should note, that in the mouth, the enzyme that work well because the enzyme that is found in the mouth, rather, it work well at an alkali or slightly alkaline pH. The pH in the mouth is slightly 7.2. As you know, 7.2 or 7 and above is alkali. Okay, so it work well at that particular pH. Okay, so the enzyme salivary amylase work well at the pH slightly 7.2, which is alkali, and that is the reason why only the digestion of starch starts in the mouth. These other food nutrients, there's no digestion. So no digestion of protein in the mouth, no digestion of lipids, only starch and part of it. Now, what happened is that after all that has happened, that is the chewing, the secretion of saliva, so through by the help of the tongue in the mouth, the rolling of the tongue as you are chewing, the food is rolled now into a mass called the bolus. Okay, so the food is rolled into a bolus, and the bolus now is ready to be swallowed. So you swallow the bolus, and this is the one that move along or the esophagus. Okay, so swallowing is just a passage of food from the mouth to the stomach, and that is through the esophagus. Okay, so that is what we call swallowing. Now, we've looked at digestion. The whole of this thing that we were looking at is digestion of digestion in the mouth. So about digestion in the mouth, it all start with ingestion, that is you take in the food. Then now in the mouth there are some secretion, of which the secretion of saliva, which contain water, mucus, and salivary amylase. You need to know the functions of these. Okay, then the tongue rolls in the mouth to form the food into the bolus, and that is what is swallowed, or something that you swallow. So the bolus move through the esophagus to the stomach by the process called peristalsis. So the process by which the bolus move from the mouth to the stomach is called peristalsis. You might find a question maybe where you're asked to define the term or to explain the term peristalsis, or rather define peristalsis. So this is just the alternative contraction and relaxation of the circular and longitudinal muscles in a wave-like manner in order to move the food along the alimentary canal. or you can say is the alternative contraction and relaxation of the sacral and longitudinal muscles in order to propel the content in one direction. So that is how the food moves. Let us look at the diagram that shows the esophagus, the cross section or the longitudinal section. So now this is the esophagus. Okay, then this part, let me say this is X, this is Y, this is Z. The esophagus is made up of set of muscles that work alternatively on yes. What are the muscles? So one of the muscles is labeled X and the other one is labeled Y. So the one that is labeled Y is the sacral muscle, or the one that from the inside part is the sacral muscle. Then the other one is the longitudinal muscle. So the inside part is called the longitudinal, the outside is longitudinal muscle. Then the inside part is the sacral muscle. And you need to know these muscles. So these work alternatively. Now the collective term that is used to refer to the muscles that work in that way, where the other one contracts, the other one relaxes, is called antagonistic. Okay, they are called antagonistic muscles. So which means when the other one relax, the other one contract. Now if you look at the Z, where the pointer is pointing, that is what we call the bolus, the mass of food. That is what we call the bolus. What is important here is that as the food is moving from this side, as it is moving going down, these muscles work alternatively, as you can see from peristalsis, in a wave-like manner, which means the other one contracts, the other one relaxes. Now it's very important that you know this is where the bolus is coming from. So we can say this is the behind the bolus. Then of course this is the ahead the bolus. So you need to know what happened to the sacral muscle, or let me start with longitudinal muscle, behind the bolus. What happened to the longitudinal muscle behind the bolus? What do you think happens? So behind the bolus, the longitudinal muscle relax. So longitudinal muscle relax. Then the sacral muscle contract. Okay? Then ahead of the bolus, the opposite happens. The sacral muscle contract. This time around the sacral muscle relax. Okay, so for the sacral muscle relax, then the longitudinal muscle contract. So these parts you need to know them as well. All right, so afterward the food has entered now the stomach, but at the entry part of the stomach there are muscles, and those muscles you need to know them. So what is the name of the muscle that is found there before even the food gets into the stomach? There's a set of muscle that is right there. What do you call these muscles? And also at the exit. So make sure that you identify these muscles. I've labeled X and W. So at the entry of the stomach we have the set of muscles, or we have muscles there. What are they called? And also at the exit, this is the stomach. So the food is coming from the esophagus, it now enters the stomach, which means we can start now discussing digestion in the stomach. I guess up to this point, with what I've explained, we are moving at the same pace. All right, so in the stomach you need to know also that it is made up of muscles. So it is made up of elastic muscles and also the glandular lining. Okay, so the stomach is generally made up of muscles. Okay, so it's just a sac made up of muscles. That's something that you should know. Then now, looking at digestion in the stomach, the first thing also you should know is that in the stomach, For digestion to occur, there is a secretion. So what is secreted in the stomach is called gastric juice. So gastric juice is secreted in the stomach. Okay, so stomach secretion, or the wall of the stomach secrete gastric juice in order to digest the food. Now listen very carefully. What are the components of this gastric juice? Okay, so the gastric juice is made up of pepsin, renin, hydrochloric acid, and mucus. And we are going to look at the function of each, like we did in the case of saliva. So, what is the function of pepsin? So, pepsin breaks down proteins to peptides. So, that is in the stomach. Now, proteins, which means digestion of proteins starts in the stomach. In the mouth, remember we said there's no digestion of proteins, but instead digestion of proteins starts in the stomach. So pepsin is the one that breaks down proteins to peptides. Then renin coagulate the milk. So this one coagulate the milk. To coagulate is to make food solid. To make milk solid. That is what we can say in terms of coagulation. So how does it make the milk solid? By converting the soluble protein, that is calcinogen, into insoluble protein, that is calcine. Okay, so this one normally is secreted in infants, and that is the reason why infant depends on milk, but of course produces solid poop, because of this enzyme is the one that coagulate or that solidify the proteins, that solidify the proteins in the milk. Okay, by converting it from soluble form to insoluble form. So this one normally in infants. Then we have hydrochloric acid in the gastric juice. What is the function of hydrochloric acid? So one of the function, I can say that hydrochloric acid has several functions anyway. One of the function is to activate the inactive pepsinogen and prorenin into pepsin and renin. One thing I'll say that renin and pepsin are secreted in their inactive form, which means the time they are secreted, they cannot act on the proteins. And we should know the reason why they are secreted in their inactive form. And that is just to prevent these proteins from digesting the origin, that is the wall of the stomach. So prevent the proteins from digesting the wall of the stomach. That is the reason why they are produced in their inactive form. One of the other function, or the other function of hydrochloric acid, is to maintain the pH of 2 in the stomach, which is optimum for the enzyme pepsin. So pepsin work well in acidic pH, that is the pH of 2. And therefore hydrochloric acid, being the strong acid, it maintains this pH so that the enzyme can work well. In addition, pepsin kills bacteria and germs ingested together with food. So sometimes we tend to eat food that have some germs, and these germs are killed by hydrochloric acid in the stomach. Okay. Then also, aside from that, hydrolyze sucrose into glucose and fructose. So these are some of the functions that these are some of the functions of hydrochloric acid. Okay, the other one that is secreted or part of gastric juice is mucus. Mucus is also produced in the stomach, and it protects the lining of the stomach against acid and pepsin. So if there was no mucus, pepsin would hydrolyze or digest the wall of the stomach, and also hydrochloric acid will hydrolyze or would damage the wall of the stomach. So to prevent that, to protect the wall of the stomach, there's production of mucus. So mucus simply protects the lining of the stomach against acid and pepsin. So this is the secretion that is there in the stomach. So in the stomach, digestion of protein start? Only proteins. Of the three nutrients that we have, that is carbohydrates, that is lipids, and also proteins. Only digestion of protein take place in the stomach, and this is because the pH in the stomach is acidic, and therefore only pepsin can work in the stomach. Very important. And in this case, the digestion of carbohydrate digestion does not occur due to the pH. Okay, so very important to understand that. All right, we continue. Still in the stomach. So after all that has occurred, the food is physically mixed. Okay, so there's also what we call churning. Churning is also physical digestion because it does not involve enzyme. It's just the contraction of the muscles of the stomach to mix the food into or result into the formation of the chyme. So churning is the one that result into that, or it's a process that result into the mixing of food. So this is just a mixing of food by rhythmic contraction of stomach muscles in the wall of the stomach to form chyme. So chyme is a paste-like, solid, milkish. Okay, so that is what we call the chyme. Then the chyme is the one that is now taken to the duodenum, which is the next stage or the next portion of the digestive system. So the duodenum is just after the stomach. So if I were to go back to the diagram, let me just go back to the diagram for the digestive system. So the outlet part of the stomach is here. So this portion here, that is where you find, somewhere here, that is where you find the duodenum. Okay, so the chyme is now taken to the duodenum. Remember, so far we've just looked at digestion of carbohydrate that starts in the mouth but doesn't end there. Then now we've looked at digestion of protein that occurs in the stomach. That is, pepsin act on the proteins to produce peptides, but of course does not end there, but it starts in the stomach. Then now we can look at now digestion in the duodenum. So the duodenum, like I said, is the next portion after the stomach. So now the duodenum received the digestive juice from the liver and the pancreas. So very important to understand here. The duodenum received digestive juice from the liver and the pancreas. What is received from the liver is what we call bile. So the liver secrete what we call bile. When the bile is secreted, it is stored in the gallbladder. Let us go a bit back, and of course I'll get to some of the questions that I've seen in an exam. So the gallbladder is this one. I'm sure you've seen it before. It contains some bitter liquids. Okay, so this is the gallbladder. So when the bile is produced by the liver, it is temporarily stored in the gallbladder. Then now the bile is taken to the duodenum through what we call the bile duct. So this part that connects the gallbladder is what we call the bile duct. So the bile duct takes the bile into the duodenum. But what is important here is for us to understand the component of or the content of the bile. What is it made of? So here we are saying the liver secretes the bile. Bile is stored in the gallbladder. You've just seen the gallbladder, and carried to the duodenum through the bile duct. You've seen the bile duct. What are the components of the bile? So one of the components of bile is sodium hydrogen carbonate. And on each we get to look at functions. So the function of the sodium hydrogen carbonate in this bile is that it neutralizes the acidic chyme and then set up the optimum alkaline pH for the enzyme of the duodenum. One thing that you should know that the enzymes that are found in the duodenum work well at the pH of 8 to 11, somewhere there. And you are looking at the chyme that is coming from the stomach where it is acidic, which has a pH of 2. So now the sodium hydrogen carbonate neutralizes the acid or acidic chyme, and therefore set up the optimum alkaline pH for the enzyme of the duodenum. That is the function. Aside from the sodium hydrogen carbonate, the bile contain biosalt. Biosalt, the function is to emulsify the liquids. To emulsify the lipids, thereby increasing the surface area for the action of lipase. Now, the biosalt from the bile emulsify the lipids. Now, what is emulsification? So, picture the large pieces of fats. Now, they are broken down into smaller droplets. That is emulsification. So, emulsification is just a breakdown of large drops of fat into smaller droplets, like I've given an example. Okay, that is what we call emulsification, and this is to create the large surface area for enzyme action. And I should mention that emulsification is also physical digestion because it does not involve enzyme. Okay, so chaining, emulsification, as well as chewing in the mouth, those are examples of physical digestion. Then the other one that we can look at is the biopigment. Now, what is the function of biopigment? Biopigment does not necessarily have any digestive function, but instead just add coloration to the feces. And that is the reason why when you eat, let's say nshima is white in color, and of course the feces comes out, it does not come out white in other words. So what add coloration to the feces is what we call biopigment, and this one does not have any digestive function. All right, so we've just looked at the secretion from the liver, which is the bile, and it contains sodium hydrogen carbonate. We've looked at the function of sodium hydrogen carbonate, and also biosalt, the function of biosalt. Okay, so the other question that we expect in an exam is that what is the role of the liver in digestion? What is the role of a liver in digestion? So it produces biosalt, it produces bile that contain biosalt, which emulsify the fats. Okay, then now let us look at the pancreas or pancreatic secretion. Since we said it receives secretion from two parts, that is the liver and pancreas. So the pancreas also has some secretion, and in this case it secretes what we call pancreatic juice. Now, what is the content of pancreatic juice? So one, it contains also sodium hydrogen carbonate, and in this case sodium hydrogen carbonate, it performs the same function as the one that is produced by the liver. So we are not going to waste time and look at the function of sodium hydrogen carbonate. We said neutralizes the acidic chyme, thereby setting the optimal pH for the action of enzyme. Then in the pancreatic juice you find trypsin, which is the one that breaks down proteins into peptides. So the digestion of proteins also in the stomach, the food does not stay there forever. So only part of it is digested, that is broken down from protein to peptide, but some of it is taken to the duodenum where the digestion of protein continues. So in the duodenum, there's trypsin that break down proteins to peptides. Then we have lipids or lipase. Lipase break down fat molecules into fat acid and glycerol. So lipase break down fat molecules into fat acid and glycerol. Now here you need to understand we are talking of three food nutrients here: carbohydrates, proteins, and lipids. The digestion of lipids starts in the duodenum and it finishes in the duodenum. Very important point. So it all starts by emulsification, where the large drops of fats are broken down into smaller droplets. Then the smaller droplets are acted upon by lipase to break down into fat acid and glycerol. And very important to know that the end product of the digestion of lipids are fat acid and glycerol. End product, we are looking at the smallest unit of fats. Okay, so fat acid and glycerol are the end product of digestion. So the digestion of lipids start and end in the duodenum. Then we have pancreatic amylase. Remember where we are coming from. In the mouth you find salivary amylase. Salivary amylase because it is found in the saliva. Otherwise, the enzyme is called amylase. Here, pancreatic amylase, which means it's produced in the pancreas, but the function still remains the same. That is to break down starch into maltose. Okay. All right. Away from digestion in the duodenum, so that is mainly what happened. Let us look at digestion in the jejunum. In the jejunum, the walls of the intestines produces the secretes the juice as well. That is called intestinal juice or succus entericus. Okay, so succus entericus is also called intestinal juice. What are the components of this succus entericus or intestinal juice? So component, it has lactase, which break down lactose into glucose and galactose. Maltase, which break down maltose into glucose and glucose. Then we have sucrase, which break down sucrose into glucose and fructose. Then we have peptidase, which break down peptides into amino acids. Now here, listen very carefully. This is the last section where digestion occurs. So digestion of the food nutrients that we are looking at, that is carbohydrates, proteins, and that is carbohydrate and protein, because lipids end in the duodenum. So here, that is where everything is completely digested. In other words, digestion ends in the duodenum. And in this case, to end the digestion, lactase digest or break down lactose to glucose and galactose. Maltase broken down or break down maltose to glucose and glucose. Sucrase break down sucrose to glucose and fructose. And these are the end product of digestion of carbohydrates or starch. So the end product of digestion of starch is glucose. Okay. Then peptidase break down peptides into amino acids. As you are aware, in the duodenum, we have trypsin that break down proteins to peptides. So here the peptides are disaccharides and are broken down to amino acids. Amino acids are the end product of digestion of proteins. Take note of that. So amino acids are the final. So digestion has ended here. Now, after digestion, what happened is absorption. And absorption now occurs in what we call the ileum, which is another portion of the digestive system. In the ileum, that is where absorption occurs. Now, after the food nutrients have been digested, they need to be absorbed into the bloodstream so that they can be transported where they are needed. So ileum carries out absorption of digestive end product of the digestive end products. And now, since it carries out absorption, by the way, is just the uptake of the soluble end product of digestion. Okay, the uptake. Now, what are the adaptations or special features that the ileum has that enable it to perform the function of absorption? So one of it is that it has thin epithelium for more efficient and diffusion of food. Thin epithelium, we are going to see that. It has a finger-like projection called the villi and microvilli, which further increases the surface area for absorption. So that is the ileum. Each villus has network of capillaries for absorption and transportation of monosaccharides and amino acids. They are very long, thereby providing large surface area for the absorption. So ileum is very long in length. Each villi has a lacteal which absorb and transport fat acid and glycerol. So these are some of the 5 adaptations or features that the ileum has that enables it to perform the function of absorption. I guess you followed up to this particular point. Then now we have this is the structure of the villus. We've heard where we are coming from, it as villus, okay, which are finger-like projection. So this is the villus. And as you can see the parts, this diagram, you need to know it. I've not seen it in an exam or in exams for a while. And these are the possible questions that you expect. Just this diagram, be able to label it, and also look at the functions of each part. So epithelium, this is the outer part. It is thin, okay, for easy diffusion of the food nutrients into the blood capillaries. Then you have the blood capillaries. Then you have the middle part, which is this part here. This in red is called the ractile. Then we have the venule this side, the arteriole the other side. Then you have the lymphatic vessel. So this one taking to the lymphatic or to the lymphatic vessel. So this is the structure of the villus. I guess given this question, you wouldn't miss it. Now, aside from, having looked at absorption, the other stage that we can look at is assimilation. So after the end product of digestion has been absorbed into the bloodstream, it is the time now they are used. So assimilation is thus the utilization and incorporation of the end product of digestion. So after the absorption, the end products are transported into the blood, to the liver by the hepatic portal vein. So that is the route that they take. So we are going to look at the utilization of each. Since end product, we are saying for proteins are amino acids. So end product of digestion of proteins are amino acids. Then end products of lipids are fat acids and glycerol. Fat acid and glycerol. Then for carbohydrates we are looking at glucose. Now we are going to look at how each of these is assimilated or used or incorporated. So let us start with the absorption, the assimilation of monosaccharides. Monosaccharides are just simple sugars such as glucose, fructose, and galactose. So how are they used? So you know that glucose, we looked at respiration. Glucose is a substrate for tissue respiration. So it is the one that is hydrolyzed in the cells to produce energy that we use for different activities. So glucose is used as a substrate for tissue respiration. So that is how it is used. Now sometimes you can have a lot of glucose in the body, excess of glucose. Where does it go? So when you have excess of glucose, it is converted to glycogen and is stored. So glycogen is just a storage form of glucose, and therefore it is stored. When it is needed at some point, it's more like you eat food, then you have some leftover, you put them in the fridge for storage. Then when you are hungry, you get them or you retrieve them, then you eat. That is what happens even to the body. So if it has a lot of energy, it uses some of the glucose there and then, but if it's in excess, some of it is converted for storage. Then if you have even more, because the body can store certain amount of glycogen, about 400 grams of glycogen. Okay? Where does, let's say you still have some excess, where does it go? So more excess glucose is converted to fats and stored around delicate organs. Okay? It is more excess glucose is converted to fats and stored in the around delicate organs. Okay? So delicate organs, we are looking at the brain, the heart, the liver, and the kidney. But also can be stored under the skin, which is called the adipose tissue. Okay? So which means this one shows that it is very possible that you can gain weight just by eating a lot of carbohydrates, because excess of it can be converted into fats. All right, I guess you've gotten something here. What I'm saying is that glucose is used in tissue respiration as a substrate, but can also be used, excess of it is converted into glycogen. Then more excess converted to fats and stored around delicate organs such as the brain, the heart, the liver, and the kidney, and also other stored under the skin. That is called adipose tissue. What about amino acid? How are they assimilated? So amino acid are assembled to make proteins required by the body. So after the food nutrients have been broken down to amino acid are assembled into the body again to form the protein required by the body. What about if you have excess? So excess amino acid are deaminated. So if you have more amino acid than what the body needs, they are deaminated. Another term that we have come across here is deamination. So you need to know this term very well, or how to define it. So deamination is the process by which the amino group on an amino acid, so the amino group on the amino acid is removed, okay, and eventually converted to urea by the liver. So amine group is something like this. So it is removed, okay, it is removed. So this amine group is removed, and that is what we call deamination. And the intermediate is ammonia, which is toxic. So therefore it has to be converted to a less toxic substance, which is the urea. But the accumulation of urea in the body is dangerous, and therefore it has to be removed by the kidney. The process called excretion, and that is one thing that we are going to, or that is one topic that we are going to cover at some point. Okay, so that is how the proteins, the amino acids are assimilated. Okay, so ammonia is the intermediate, during which deamination and the highly toxic, so it quickly converts to urea. I think this is something that I'm from explaining. So now, what about fat acid and glycerol? How are they assimilated? So glycerol and fat acid are assembled or combined also to form fats. So that is how they are used. They are combined to form body fats. And of course, you need to know the function of fats. What are some of the functions of the lipids in our body? So one of them is for insulation. The other one is the source of energy when they hydrolyzed. So quite a number of them. Get to look at some of the functions of the lipids in our body. Otherwise, we can move to the other portion or the other section of the digestive system, which is the large intestine. The large intestine, the function of the large intestine is to carry out absorption of water from the feces, okay, or undigested food called feces. So absorption of water from the feces, that is the main function of the large intestine. Sometimes they will tell you the colon. Okay, so very important to understand this particular part. Now, what is the function of the liver in digestion? I think I explained this one at some point. What is the function of the liver in digestion? So for me, I would say it produces bile salt, which helps in the emulsification of lipids. Do you have another function of the liver in digestion? Get to contribute as well. So make sure that you type in your answer in the comment, and we'll be able to learn from you as well. So what is the other function of the liver in digestion? Please, strictly in digestion, not the function of the liver, but strictly in digestion. So that is very important to look at. All right. The other part that you're going to, or that we can look at, are the ailments of the liver, the disease of the liver. So we have what we call hepatitis, and this is just the inflammation of the liver caused by the hepatitis viruses. Then we have what we call cirrhosis, which is the hardening of the liver and resulting from excessive intake of alcohol. Okay, what are some of the ailments of the liver? So get to also contribute on this particular part. The other part that we can look at are the ailments of the stomach, and of course this one I'm going to leave it up to you. What are some of the ailments of the stomach? These are very important. If you check questions, you find questions on this. So on this part is very much examined. Make sure as you study, you don't forget to answer these questions. All right, so we've come to the end of the lesson. If you've enjoyed the lesson, don't forget to subscribe to this YouTube channel and make sure you turn on the notification bell so that whenever I post the new materials, you'll be notified. If you are interested for online lessons and also physical lessons, for physical lessons, we are found in Kawata, that is Lusaka, near Kawata Primary School. But if you are interested in the online lessons, you can get in touch on the number 0976 402563 and we'll be glad to have you in class. The earlier you start, the better. Let us prepare you for distinctions. Otherwise, we can end here and see you in the next one.